So the next process that we'll be talking about is called transcription. If you remember back to the warm-up in class, we talked about transcribing something is to copy something. So what we'll be doing in this process is copying an important section of DNA into a section of RNA. Now, if you look at the two structures, there's some very important differences to recognize. For starters, DNA is double-stranded. So there's going to be a strand on both sides, which makes DNA a lot more stable than RNA. Another important difference involves the different nitrogenous bases. DNA has A, T, C, and G, which we talked about earlier. Most of those are present in RNA, except with one difference. It has uracil now instead of thymine. So this is going to be one of the very important differences between the two molecules. Those are the two main things I need you to know, that RNA is single-stranded, which makes it a little bit less stable. RNA is usually only good for a few hours in the cell, and then it starts breaking down to a point where it's unreadable, whereas DNA is very, very stable. Obviously, many of your cells have DNA for decades, and it's a stable molecule. It's not something that's breaking apart very easily. So single-stranded versus double-stranded, uracil versus thymine. The next thing we have to talk about is why we even need RNA. And ultimately, it comes down to where the DNA is located. If you remember when we were talking about the parts of the cell, the DNA is located in the nucleus. The DNA is extremely valuable to the cell, and because of that, it has to be protected there. So the DNA isn't used directly. If the cell needs information from the DNA, that information is transcribed, it's copied onto a section of RNA, and then that RNA is used in the cell for information. Most specifically, most of the time, it's sending that information to the ribosomes in order to build proteins. So now that we've established why we need this one, we're going to take a moment to look at the actual process here of how this works. I'll get these guys out of here. So when it comes to transcription, there's something important involved called RNA polymerase. This is an enzyme. It's shown here, and it's the enzyme that's taking the strand of DNA. You can see the DNA splits open, it like unzips in the middle like a zipper on a jacket, and then each side is copied into a section, or actually one side, I should clarify that, one side is copied into a section of RNA. You can see things still bond up the same way they did during replication. So in this case, we've got the blue and the red ones together, so that's cytosine and guanine pairing up, although now we have a slight new introduction. You can see the book kind of colors these things similarly. Thymine is a little bit of a lighter yellow, uracil is kind of like a darker yellow. So in this case, we only have uracil, this dark yellow one, shown in the RNA, whereas the light yellow thymine is shown in the DNA molecule. Another thing to keep in mind is the whole DNA is never copied. RNA is just one small section of it. So you can see we've got extra DNA on both sides, but just this one little section here is being copied into a strand of RNA that'll be used somewhere to make a protein. So again, one of the important things to get from this is this guy called RNA polymerase. That is the enzyme that's actually copying this. That's what's sort of shown here, this big blob. DNA polymerase does the same thing when it comes to replicating a section of DNA into a section of RNA. So the last thing that we have to talk about as far as practicing this is what to do if I give you a section of DNA that you then need to copy into a section of RNA. So the trick with this goes back to something very similar that we did when we were going through the process of replication. You have to write down what pairs together. So we still have C pairing with G, but now if you recall, there's a slight difference with this one. We no longer have thymine. So adenine now pairs with uracil. A way to remember this, because I sort of gave you the trick that C and G are of a similar shape, A and U are both vowels, so that's one way to kind of remember that those two go together. So now if we're copying a section of DNA into RNA, adenine, the A, if we go back to our little cheater thing here, A is going to pair up with U, second A pairs up with U, C pairs up with G, G pairs up with C, C pairs up with G again. This is the one that throws everybody off. Remember, adenine is still fine. So there's thymine in the DNA section that still pairs up with adenine in the RNA. But it doesn't work the other way. There's no longer a thymine down here. A, again, now pairs with U. So just remember, the vowels. Vowels are going together. So C then pairs with G, G pairs with C, and then lastly T going with A. So just like replication, once you get this down for just a couple of a base pair long sections, whatever this one is, it's like about 12 or 13 base pairs long, you could do this for any length of DNA. The main thing to remember is just what pairs together. So you associate C with G because of their shape, A with U because they're both vowels. You could now copy basically an infinitely long section of DNA into a section of RNA. And remember, this RNA molecule is going to be going out into the cell to be used for information. So most likely it's going to be headed to a ribosome where it's used to make a protein. So the next video actually involves that process. It'll be called translation, which is the process of taking this RNA molecule and then using it to build a protein at the ribosome.